Okay, so now we're going to start with the last section of chapter 4. So we've almost made it through what I consider to be a pretty content-rich chapter and a pretty difficult chapter. Before this, we were speaking about DNA. So you have DNA right here, and we were talking about DNA, and we went through, the shape of DNA, that it's made up of nucleotides, right? And each nucleotide is a phosphate, a sugar, and a nitrogen base. And the only thing that's different in the nucleotides would be those 4 nitrogen bases. You have A, T, C, and G, right? And A always bonds with T, T always bonds with A, G, and C, and so on down the line. So you always have this base pairing rules going on across the line here. So if this is an A, this is a T, if that's a G, this is a C, or whatever down here. All right? And those are held together by hydrogen bonds. Remember, the hydrogen bonds are pretty weak bonds, so they can break apart and you can go through replication. That whole process we talked about, copying DNA, all that is really being able to put a set of recipe instructions in every new cell. Not every cell is going to express every single recipe in that book. As we look at the entire genome, a stomach cell is only going to express a small set of those genes, only the ones it needs to produce hydrochloric acid or whatever it's needing to produce. Your cells that make melanin for pigmentation in your hair, your skin, your eyes, whatever, they only express a very different small set of cells. This is what we're talking about with the expression. The expression is this ability to make proteins, so protein synthesis. DNA is just the recipe book, okay? So it's not doing anything with it. I can have an entire book in my kitchen, but unless I do something, I take one of those recipes and I make cookies from it or whatever I'm making from it, I don't get anything. I can't eat the book, right? I can't do anything with the book. It's just a code that says this is how I want you to do something. That's what DNA, merely the book. And replication is just Xeroxing that book or going through a printing press or something and making a copy of that book. It's not really putting that book to work. So what DNA really has to do is it has to take that recipe, it has to take that code, and it's eventually going to have to produce a protein. And so in order to do that, it has to copy the recipe down first. So what it does is it makes this code, or it copies its code down into something we call RNA. And we have steps here. Step 1, doing that process of making RNA, we call transcription. Then that copy of just that one little recipe, not the entire book. So DNA is the entire book, right? In my little analogy, mRNA is just one recipe out of the book, and you just made a copy of it, okay? Then that one recipe then can actually be used to make the protein. So now we're making the cookies, or you're making the cake, or whatever it is you want to make here. So you're actually making something that's useful, the protein. And our protein is what we refer to as a trait. So if I was talking about the melanin cells, that would be your hair color, right? So that's the trait that we see. So the job of DNA is to hold the code. The job of this other player that we're going to introduce, RNA, is actually to put it to work. It's the cook. It's the one that's going to actually do what you need to do to get something useful from that code. So we do have those 2 steps. First, like I said, transcription is making the messenger RNA. We're going to talk about it first. And then translation is taking code. So this is all code, right? This is code. We made another code, and then we're actually translating it into something useful. And useful is the trait. So something that the cell, something that the body can actually use. So let's get started on that. So our genetic code, remember, is just a set of instructions. What it's instructing the cell to do is put amino acids into the correct sequence, the correct order. So if I have, there's 20 different amino acids. So we have 20 different types of amino acids, right? And if I have, and I'm going to make this up, this isn't, something I'm giving you the code to some specific protein, but if I have one that's like valine and alanine, serine, and threonine, right? Each one of these is an amino acid. The order of these, valine first, then alanine second, then serine third, and then threonine fourth, that is coded for by the DNA. So the DNA says, I want you to put these in this specific order to make this specific polypeptide, which will become that protein. So remember, when we talked about the structure of a protein, the first level is the correct order of those amino acids. And then the second level, if I get that order correct, and I have valine, alanine, serine, and threonine all in the correct order, plus 200 more of these, then it will start going into the secondary structure correctly, those alpha helices or beta pleated sheets, and then it can fold into the tertiary correctly. It's all a domino effect. And then with those four, van der Waals forces, ionic, hydrogen bonding, and disulfide bonds, and then it can form your actual protein, which is your quaternary structure. So it takes the first step, putting these amino acids in order to actually produce the correct protein. And who holds that code? DNA. But DNA just holds the code. It doesn't actually bring in amino acids. It doesn't link them together with bonds. It doesn't do any of that. It just holds the code. So what we need is another player. We need something else to come in that can actually put that together and can actually start that building process. But we're going to have to define some terms. So in my sequence of DNA, so I'm going to draw out this little line. Let's just represent that as the phosphate sugar backbone. I'm not going to draw those out. And then I'm going to put these little letters on here representing the nitrogen bases, right? Each of those, remember, you're going to have a double-sided ladder. We're just, for a protein, all you have to do is use one side of the ladder, so like this side or the other side, but not both. So we're going to take this ladder, we're going to separate it, and we're going to have access to those nitrogen bases in there. Each set of 3 nitrogen bases is what is going to be read to code for one amino acid. So like ACC might code for something, again, I'm making it up, don't quote me on it, like the first one, valine. And then GAT would code for the second one, alanine. And then C, whatever it is down the line, serine. So it's each group of 3 that's coding for one specific amino acid, all right? So we refer to those triplets as codons. You refer to it as a codon on DNA, and we're going to refer to it also on the messenger RNA as a codon. Code on, all right? So it has the code. All right, so since I talked about RNA being the actual worker bee, RNA is going to be the cook, the thing that actually makes something out of that recipe. So he's our cook, right? Keep up with that little analogy. Let's talk about what it looks like. So remember DNA. So here's DNA over here, and it's this double-stranded ladder, right? So it's a double helix. That's DNA. RNA is not. RNA only uses one side of this ladder or the other side of the ladder, so it really only has to copy one side. It only needs to be a single-sided ladder. So instead of this double twist, you only use one side, or you use the other side, but we're not using both. DNA gets its name from the sugar deoxyribose. Well, RNA gets its name because it uses a different sugar. It's going to use ribose instead of deoxyribose. So that's where the R in RNA comes from. Remember, the nucleic acid is Na. DNA is trapped and stuck and cared for in the nucleus. So it's kept intact because it's really important. You don't want to destroy the original recipe book or you'll never have the recipes again, right? So it gets protected in the nucleus and it has to stay in the nucleus. Unlike that, though, RNA is a little more stable, and RNA can be sacrificed. I could get another cook, right? So you can make a new one here. So RNA actually has the ability to go from the nucleus out to the cytoplasm where it would need to hook up with a ribosome. Remember, our ribosomes are the organelles that make protein. So if I'm going to make protein, obviously I'm going to have to involve them. So RNA will be made in the nucleus. So it's made here. And then it moves to the cytoplasm. When it moves out to the cytoplasm, it's going to find a ribosome, and it's going to hook up with that ribosome. Literally, they join together. The ribosome and the mRNA join together. Nucleotides in DNA have the nitrogen bases A, T, C, and G, right? Well, RNA pretty much is close to that. It has A, it has C, and it has G. Unfortunately, thymine is not very stable in the cytoplasm and will tend to break apart, so it's not a good player to use here. So we replace it with uracil. So you still have A, you still have C, you still have G, but now we have a new player, a substitution called uracil. We're still going to have the same bonding. Since C and G are still involved in RNA, you're still going to have bonding with C to G or G to C. A is still available, so it's going to be seen in RNA too, but where I would normally find a T, T has been replaced by uracil. Now keep this in mind. If you see a sequence, I give you a sequence like this, and I say, okay, is this RNA or is this DNA? You know automatically from those T's that that is DNA. So this would be DNA. But if I give you a sequence that looks like this, oops, sorry, let's put you there. Then you're going to say, oh, that's RNA because it has U's. So look for those letters. Look for those clues that tell you which one's which, all right? RNA has ribose sugar. DNA has deoxyribose sugar. RNA has AC, we'll put a G on here too, G and U, whereas DNA has A, T, C, and G, right? The T's and the U's are swapped out for those two. DNA is a double ladder, RNA is a single ladder. So those are differences in there. So when we're looking at this, okay, remember that only one side of the DNA ladder is used. So when you see DNA here, it would normally have a second side to the ladder, but we're not using it to make protein. So that DNA ladder untwists, unzips for a little while, and that gene is exposed, and that G can make an RNA. So here's my phosphate, here's my sugar, and in DNA that would be deoxyribose sugar, right? And there's my A, T, C's, and G's. In RNA, you're going to have a phosphate. You've got a sugar, but now it's ribose, right? And you're going to have A, C, G, but we've substituted U here. So G and C are still going to bond together like they normally would. CG, GC, that's no change there. But with DNA, it's A to U. And if I see a T, I know I've got DNA, right? That doesn't change because that's DNA. But it's going to bond with an A over here in RNA. It's just when I see an A, will I see a U in RNA. So I can tell a couple things from this. If I see the T's, I know that this sugar is going to be deoxyribose, and I know that originally it's double-stranded, right? And I know that I've got DNA. If I see a U right here, I know that my sugar is going to be ribose. I know it's going to be always single-stranded, and I know I have RNA. Otherwise, bonding and everything's still the same. So there are, unlike DNA, where your DNA generally you have one type, or at least one type that you guys learned about that you need to be responsible for. There really are multiple types of RNA. So there's the main player, the mRNA, then there's these secondary players that are going to come in and help. So like in a restaurant, you have the head chef, you have the sous chef, you have pastries or whatever. You have all these other guys that are helping out, and guys and girls that are helping out. So the same thing's going to happen with RNA. So there's different types. We just label them with a small letter out in front of the 3 letters of RNA. The first type, the main type that we're going to start off with, is called messenger RNA. That's where the M comes from. So the M comes from the term messenger RNA. Messenger RNA is directly made from the DNA. So where I have DNA right here, DNA holds the code. This DNA ladder that you see in this picture is going to separate. So those 2 sides of the ladder will separate. Only one side of this ladder will be used to make that messenger RNA. So the code down one side as we go straight down, so the A, T, C's, and G's as we go down the ladder, right? Only one side is used to form the other side for that, or that side for the RNA. All right. And it's the guy that can now leave the nucleus, go out to the cytoplasm, hook up with a ribosome, and begin that process of making proteins. This process where we make RNA from DNA, that process is called transcription. That's our first step to making a protein. You have to transcribe, that means write down, an RNA molecule from DNA, and specifically it's messenger RNA. Transcription, transcribe just means write down. Write down the code, take the code and go put it where it can be used. Take it to the stove. All right. Remember that RNA is a single polynucleotide, so that's not any different from your mRNA to any other RNA that I'm going to introduce here. It's still complementary. With DNA, the complements were A to T and C to G. In RNA, it's going to be A to U, C to G. So we have that substitution right there with the U. So here's my DNA ladder. Notice it's double-sided here. It's split apart where you see this pink square, and only the one side is being used to make the messenger RNA. So where I have an A right here on DNA, I'm going to have a U nucleotide that comes in. Where I have a C, I'm going to have a G come in. Where I have a G, I have a C, A to U, A. And here, where I have a T, it's going to be an A on messenger RNA. So the base pairing rules still apply. It's just we have one substitution. In the replication of DNA, when I made another exact copy of DNA, we used an enzyme I told you need to know called DNA polymerase. Now I'm making RNA, so I'm still going to use a polymerase, but I'm using specifically RNA polymerase, and that's what you see in pink right there, is the RNA polymerase to make that messenger RNA. Second player, tRNA. So messenger RNA gets made in the nucleus and then travels out to a ribosome in the cytoplasm. Once it hooks up with the ribosome and they come together and form a unit, then it's time to bring in the amino acids, and that's the job of the tRNA. So T stands for transfer. tRNAs look vaguely like a T. Some people, they also refer to it as a clover leaf. If you look at the first picture, you can see this T shape. It bulges on the edge, so that's where they say clover leaf, because it has three bulges on three sides. Really, in reality, it looks more like this guy right here. It's wound up, and I'm not really sure where they get that idea from, that it looks like a T, personally. But there's two important parts here. First of all, it is a single, just like messenger RNA, it's a single nucleotide, polynucleotide that goes around like this. So it has a beginning and it has an end. It has two ends to it, this end right here and this end right here. The first end up here that you see that really shows nothing except that ACC, this is where you would have an amino acid attached. So that's the attachment for an amino acid. So where I was talking about valine, threonine, leucine, whatever, that would be attached there. One of those are 20 amino acids. And those 20 amino acids, one of those is very specific to a type of tRNA. So, you know, one type of tRNA, one amino acid, another tRNA, another amino acid. The other end is what we see down here on the opposite side, okay? So that end is what we call the anti-codon. Remember, a codon is a group of three on DNA, or we're going to also refer to it on messenger RNA, so something like this. So this would be one long strand. Let me put them all together. U, C, C, G, G, A, U. Okay, so this is my messenger RNA. I know it's RNA because you see the U's right there, right? First thing I always want to do is divide it into the groups of three. So there's my groups of three right there. Each of those three messenger RNA is called a codon. My tRNA, right here at the end, there's a group of three that will correspond to that messenger RNA. So where ACC is, when my tRNA comes in and brings in its amino acid, there's going to be a U, G, G on this little section right here that bonds with it. So this code ACC that corresponds to the code that came from DNA, right, is specific to an anticodon UGG, and that anticodon is specific to one tRNA that carries one specific amino acid. So you could see at this point where the DNA is specifying a specific codon on messenger RNA. That codon on messenger RNA is specifying a specific anticodon on a tRNA that is bringing in a specific amino acid. That's how it knows what order to bring the amino acids in. And then UUU, your tRNA that would come in would be AAA. CCG codon would correspond to the anticodon GGC. And then CAU would correspond to GUA anticodon. So these are the anticodons right here on tRNA. So messenger RNA carries the code, hooks up with a ribosome, right? tRNA brings an amino acid, and it's the codon to anticodon hookup that's important there that brings in the right amino acid. This is what we call step 2 in making the protein. This is translation. Step 1, transcription, make the messenger RNA. Messenger RNA leaves the nucleus, goes out to the cytoplasm, hooks up with a ribosome, and then that begins the next step, step 2, translation, the actual production of a protein by bringing in the amino acids in the right order. Then we have a third guy, rRNA. That stands for ribosomal, and really the only thing you need to know about that is that rRNA makes up the ribosomes. Okay, so the ribosomes themselves are a RNA component. They also have some protein in there, but they're made up of this substance called rRNA. So we have 3. Messenger RNA made in the nucleus by DNA, tRNA brings in the amino acids, transfers, and rRNA that makes up the ribosomes. And look here, when you look down, let's look at this picture down here. Here's the messenger RNA. Told you it had to hook up with a ribosome. The brown is the ribosome. There's 2 little units that hook together. And then there's 3 spots. These blue cylinder looking things, these are the tRNAs. And each one of these little purple things is one amino acid. So this tRNA that's right here is going to specify only a specific amino acid. This one specifies a completely different one. And it's the job of the ribosomes to start hooking those amino acids together. So you can see that this chain of amino acids, there were tRNAs that brought in that one, and then brought in that one. And once they get hooked together, the tRNA just leaves. It's done its job. Okay, so first step, transcription. Here's my DNA in blue. DNA separates. I use one side of the ladder to bring in my messenger RNA nucleotides to build my messenger RNA. And remember, that is called RNA polymerase. That's the enzyme that actually allows that to occur, all right? Occurs in the nucleus because where's DNA found? DNA is found in the nucleus, so it has to occur in the nucleus. Then that messenger RNA that you see in pink is going to move out to the cytosol to find a ribosome. This whole process of making the messenger RNA, that's transcription. Translation, step 2. Messenger RNA that's already been made now leaves the nucleus, goes to a ribosome where they hook up, join together. That's the beginning of translation. Then tRNA is going to come in and bring in the correct amino acid. So here, where I have, here's my messenger RNA, and it's one single strand, and here's the code. First thing you always need to do, come in and make lines between the codons. Just draw them in or do something that allows you to see them in groups of 3, right? So just visualize groups of 3. Makes it easier. Step 1, remember, making the messenger RNA. This happens in the nucleus. So we already did this. where I have A, C, C on my DNA, A bonds with U, C bonds with G, C bonds with G, A bonds with U, A bonds with U, A bonds with U, C to G, C to G, G to C, A to U, G to C, and T on DNA to A on RNA. So there's my small section of messenger RNA that's been made. Messenger RNA goes out to the nucleus, hooks up with a ribosome for translation to begin, and then tRNA is going to bring in these little amino acids. So each of these little shapes is one amino acid that is attached to a tRNA. So the tRNA then has to bond with this code that you see on messenger RNA. So once again, the tRNA anticodon, so this is called a codon. Each of these groups of 3 is a codon, right? Those groups of 3. These codons have to bond with an anticodon on tRNA. So the anticodon is going to be specific. So if I have a U right here, the anticodon would be A, G to C, G to C. So my first anticodon, UGG codon, corresponds to anticodon ACC. The next codon is UUU, so what is my anticodon? My third codon is GGC, so what is my anticodon? And then my third codon here is UCA, so what is my, fourth, is UCA, so what is my fourth anticodon? U to A, C to G, and A to U, okay? And then those anticodons are bringing in, are on a tRNA that's bringing in a specific amino acid. Now I have my primary structure of a protein. All it has to do from now on is to fold and join in with other polypeptides, and you get a protein. Okay, so here is in brown, that's the ribosome. 2 subunits, large and small, came together. In pink, I have my messenger RNA, and you can see the codons, so go in there and mark off your codons, right? The first tRNA that came in, my codon was UGG, so the anticodon is right there, it's ACC, and it brought with it an amino acid. Once it gets in that spot and the next tRNA comes in with its anticodon matching up, bringing in its amino acid, so phenylalanine belongs to this one, tryptophan belongs to that one, there's a bond that forms between them. And it's called a peptide bond. And when that occurs, that tRNA releases this. So the reason these two are not attached anymore, this one and this, and tryptophan right here are not attached, is because we formed a bond right here. So the tRNA released it, it goes back out to the cytoplasm. It's going to pick up another one and come back later. This tRNA here, number 2, is in this spot. So number 3 comes in and joins in with the next codon, and it's bringing with it attached its little amino acid glycine. And when that comes in, then we'll form a bond between phenylalanine there and glycine. And then number 2 will release its amino acid, and everything shifts down. And you continue reading down that messenger RNA, moving down that direction. All right? And what you're left with in the end is a long strand of amino acids, which is your primary structure of your protein. The tRNAs are reusable. They're like taxi cabs, like I talked about with cellular respiration. These guys are also like taxi cabs. They pick up a amino acid, they bring it to the ribosome, they drop it off in the right spot when they're directed to, and then they leave and they go get another one. So it's a very orderly process. What is all that code? How do I know? Let me go backwards. How do I know that this tRNA right here carries glycine? because the code, the anticodon and the codon specifically, it always goes back to the messenger RNA, by the way. The messenger RNA codon has a code that says you can only bond with this tRNA. So this code right here can only bond with this tRNA. This tRNA can only carry that specific amino acid. So somewhere along the line, people all worked out, the scientists worked out the different codes that represent each amino acid. And all life has the exact same code. So it doesn't matter if I'm talking about your cells, a plant, a fungus, or a bacterium. We all use the exact same code. So that's pretty cool. That means you only have to have one chart and you can use it for everything. Okay. The code is also what we call redundant. If you look here, let's look at this one, this section right here. This is all mRNA, by the way. These code charts are called codon charts. So they're actually referring to the messenger RNA, not the tRNA, not the DNA. So always make sure that you're looking at the messenger RNA. But if I look at these codes right here that I just squared off, UCU, UCC, UCA, and UCG, all of those codons really specify the exact same thing, okay? So we have 20 different amino acids. Just write it here. 20 different amino acids, but we have 64 different codes that could be used. That means we overlap some. So here, proline, you can see that proline has 4 different codes for it. And then leucine actually has 6 different codes. So there's multiple codes for each amino acid for the most part. The only one that doesn't is this one right here. AUG only specifies methionine, and it also is specific. It specifies the start. Every single protein, the codon that begins it is AUG, which means start, but it also means methionine. So every single protein starts with an amino acid called methionine, which is nice because remember our DNA? Your DNA is this long strand of gene after gene after gene after gene after gene. So gene 1, gene 2, gene 3, gene 4, right? So on down the line. If there was no start and no stop, you would just continue coding every single time all the way down the chromosome. So what we want to do is we want to say, hey, this gene starts here, start codon. This gene stops here, stop codon. And then you would have a new start and a new stop for the next one. So it specifies the beginning and the end for each gene. Again, the universal start is AUG. There's 3 actual stop codons. So anytime it comes across any one of these, it doesn't have to be, it's not going to be all of them, it's just going to be one. So UAA, UAG, or UGA, that says stop, we're done. Okay, so there's always a start and there's always a stop to every protein that's being made. Can't stress it enough. Even though you have this DNA code, you're going to see, you're going to have an RNA code that's messenger RNA, you're going to have a tRNA anticodon. I want you to be able to do those things and to tell me what letters they would be, but your chart is always, always, always the messenger RNA, okay? All right, so there's my start. I circled it. Forgot I did all that. And here's my stops, okay? And then everything else, these are all necessary amino acids. Their order is going to determine the order of the structure of your protein. All right, so start here. So here's my DNA. Remember, it's going to be read in groups of 3. So as we come down, we're going to look at it in groups of 3. So there it is right there. DNA, if I asked you to make the other side of the DNA ladder, okay? So if I say in the test, make the other side of the DNA ladder, what would bond to this? That's where you would go A and you would use T's, right? T to A to T, C to G, G to C, C to G, A to T, G, T, A. You need to practice this, by the way. Don't just watch me doing this and assume that you're going to remember how to do it on test day. So make sure that you are practicing, and I've got some links in D2L for you to go in and practice their click and drag things, so to practice with that. If I asked you to make the other side of DNA, you're going to use the T's, right? But if I say, I don't want you to make the other side of the DNA, I want you to make a protein, then what you need to do is make your messenger RNA first. So we want to make the messenger RNA. That means T in DNA bonds with A. A is now going to bond with U, C to G, and so on down the line, right? A to U. So remember that we're making that substitution. If I tell you that you're transcribing your RNA, transcription is RNA. Replication would be the other side of the DNA. So keep those in mind as you start working through this. Keep your terms straight so you know what you're supposed to be doing. So here's my messenger RNA. So the T and the A, right? The A to U, the C to G, and so on down the line, right? So if I just corresponded that, that would all match up. Remember I told you AUG is the universal start? So every gene is actually going to start with the DNA TAC, so it can make the messenger RNA AUG. And then those groups of 3 are going to specify, if you use that little chart that I showed you, you don't need to memorize the chart. Please don't memorize the chart. I don't know the chart. I know some things on the chart, but I definitely don't know all of it on the chart. What you need to know how to do is use the chart. So we have to use the chart. Okay, going back here, my first base here is the first right here. So you would find the U, C, A, or G. So if I gave you a code, let's practice. If I gave you the code AUG to look up, your first base, you look down this side right here. So I come down and I find my A and say, okay, there's A right there. So I know I'm in this row and I've pretty much eliminated everything else, okay? My second code here is U. So I come across the top where you find the second, and again, you have those 4 letters, U, C, A, G. I find U, and I know that I'm in this column here, so I've just eliminated all of this. The only thing I have left is this little box, and then I find the G, and I've got it. Third base is over here. All right, so I'm going to give you a second to think about it. But if I give you a code that's CCU, you need to look that up and tell me what you get, or think about it, and then I'll tell you if you got it right. First base, second base, third base. So C, we're here. C, we're here, right? And then U right there. So there it is right there. And my amino acid is proline. Okay, give you another chance. Let's do UCC, right? Use the first letter, so I'm right here. I'm in this row. Come across, C's right here, so I'm in this column. Use C, and the last one is C. It's right here, so my amino acid is serine. And this works for everything, okay? So when we come across to this, and I've got this right here, this AUG, you would just go to that chart, look up your messenger RNA and say AUG corresponds to methionine, and you would look it up. CGU, first base, second base, third base, look it up, and it's arginine. GUA, valine, and so on down the line. You just need to write down whatever, or specify whatever's there. Okay. I've got it color coded so you can see the colors correspond. Draw out the lines, do something, whatever you can do to kind of make sure that you see it in groups of 3, because it's really easy if it's not color coded, if it's not lined up. It's really easy to be going along here and then get off, because your eyes get that drift going on. So the first thing you need to do is kind of make some sort of notification to yourself that you're dividing it so that you can see it. and you stay there. So you get too far down the line, it all starts to get run together. All right, so sort of in summary, here's my DNA. DNA opens up, only one side is used, so in this case it's this side that's being used. You make a messenger RNA. That whole thing's called transcription. By the way, if you're trying to remember what comes first and what comes second, C in transcription comes before L in translation, right? It's just one way to do that. Transcription, and it occurs in the nucleus because that's where DNA is found. That messenger RNA will unbind from that, and there it is unbound right here, and it moves out of the nucleus, finds a ribosome, which is made up of rRNA. They bind together, and then your tRNAs will come in, and your tRNAs bring with them specific little amino acids based off that codon chart using the messenger RNA. And that specifies a specific sequence of amino acids, right? So that's specifying one sequence of amino acids. This whole process is called translation. I'm translating a code into something useful for my body, a protein. All right, told you there's a bunch of different RNAs, and there's actually some different types of DNAs too, that if you get into molecular biology, you'll learn more about that. But for our intents and purposes, we only need to know about the one DNA, and then the two or three, I guess I should say, messenger RNA, tRNA, and rRNA. That's part of the transcription translation process. And then there's some others that we're constantly discovering. We don't know everything yet. One group is called what we call microRNAs. And what we found is microRNAs are made, but they really don't code for a protein. Their job really is to regulate things. So they go in and they regulate whether a gene is turned on or turned off, and they're regulating that whole process in there. So we can look at this and we can say, how about if we make our own synthetic RNAs? RNAi is a synthetic form that's based off this ability of our cell to make these microRNAs. And then we can say, block this gene expression, which means turn off this gene if it's not behaving properly. If it's not doing the right thing, let's just shut it down and keep it from happening. In cancer, remember, cancer is cells going through too much copying, too much replication, out of control. This is a way to just turn those guys off, possibly. So that would be a way that maybe we could treat genetic disorders, or we could treat things like cancer, is by using these RNAis to block the transcription of a protein, translation of a protein. Your DNA goes through replication very, very quickly. Remember, the RNA polymerase brings in those correct nucleotides at the right spot, but it does it at a very, very fast rate. We really want to get that made and then go through mitosis and then get back to business again. So it goes through it really quickly. So really, we do actually have error rates in there, but the RNA polymerase and some other enzymes have the ability to go back in and spot check things and try and correct it. So when we look at the statistics here, I told you, you're mostly the same as every other individual. Only about 0.1% is actually different from the person, next. And I'm not talking about your family members. I'm talking about just the general public. Only about 0.1% of your DNA is actually different from person to person. That means 99.9% of your DNA is exactly the same as every other human beings on this planet. You remember, you're 98% the same as a chimpanzee, and there's a huge difference between us and them. So that tells you how much has to be the same. And that's just because most of your DNA is coding for enzymes that are necessary for life, and specifically for human life in this case. So when we go to find this thing right here, is called a DNA fingerprint. When we look at a DNA fingerprint that is unique to just you, we are targeting that 0.1%. that part that is unique to you. And we look at it and say, if this corresponds to you, then that is the only part of your DNA that is different from everybody else's. Mutations occur when there's errors in replication. So your DNA went through its normal copying the DNA, splitting, and replicating, but now there's been an error. Okay, that error usually gets checked. DNA polymerase and some other enzymes usually go in and they'll check for those errors, but sometimes they go unchecked and they're left alone, and that's a mutation that then could be passed along if that happens in the gametes, which are your sex cells. So we changed the order of the bases by accident. So like here, I've got the order should be TTC, but accidentally during replication, an A was brought in. Normally that would be rectified, so you would have DNA polymerase or another enzyme come in and cut that out and repair it. But every now and then it goes without being checked. This is called a substitution. So the T got substituted with an A. So we do have some errors, mutations that are called substitutions. Then you can have some that are additions or deletions. So what happens when this process of replicating this DNA right here, you actually got an extra one substituted in. So this was correct, so that's supposed to be a T. TTC was correct, but accidentally you got an extra in there. That would be an addition. If it was going along and this one got left out, so I went from T to C, that would be called a deletion. These are really destructive because a substitution right here, this right is going to code for one amino acid. Now I have a different amino acid, so where it was glutamic acid, now it's valine. That's a different amino acid, but every other amino acid in that chain is going to be correct. So you're changing one amino acid, we're not changing the entire protein. But if I have an addition right here, I've just changed my reading frame all the way down and it becomes much more destructive. So from a wrong protein to a junk protein, there's a big difference in there. And I'm not saying that this is minor here. Going from the glutamic acid to valine, or going from one amino acid to another, only changes one amino acid in a series of like 200 amino acids, but it can be a really big difference. One of the most common genetic disorders in African Americans comes from the west coast of Africa, and it's called sickle cell. Sickle cell disease is where we should have these red blood cells that are nice and round, and they carry a lot of oxygen. But in sickle cell disease, because of this substitution right here, you get a cell that will sickle in shape, and it doesn't carry much oxygen. It tends to get caught in capillaries and cause blood clots, and so you end up with tissue damage and organ damage. And if it goes unchecked, it can be very destructive to the individual. And in countries where the medical community is not very stable, they don't have a good healthcare system, this is the number one cause of children by the age of 5 that's not infectious, by the way. That's a genetic inherited disease. In Caucasian Americans, you have one called cystic fibrosis. So this is sickle cell. We have one called cystic fibrosis that originated in Northern Europe, and that is a substitution in here. Actually, it's a deletion. It's a deletion of a group of 3 nucleotides. So you go from, instead of substituting one, we go from a sequence, say it's a sequence of a T, I'm going to do the TTC, and then a CG, right? This was completely eliminated, so that was a deletion. So this is read correctly, this is read correctly, but you're skipping an amino acid here. So if I had valine here, let's just go with glutamic acid. Glutamic acid right here, and then I had lysine here. Whatever was supposed to be here is no longer there. So you're missing an amino acid. And again, that causes the incorrect shape. And what that causes in the individual is a buildup of this very thick mucus in the lungs and the pancreas and other organs, and it causes them to basically suffocate on their own mucus. They can't exchange oxygen, carbon dioxide, and they suffocate on that. The thick mucus that collects in the lungs can also cause a lot of bacterial infections. So you see a lot of bacterial infections in the lungs, like pneumonia and stuff. And again, untreated, unchecked, this can be death at a very young age for these individuals. So these mutations, whether it's simply a substitution, can have devastating effects like sickle cell, or if it's a deletion or a multiple deletion, can have devastating effects like cystic fibrosis. Or it could be even worse, and that individual could never even develop as a fetus because something so major was incorrect in the development of that individual. So let's look at some of these. First one, a point mutation called a substitution. Remember, it's just the substitution of like one nucleotide. So here's my original DNA: TAC, GCA, TGG, ATT, and in this little diagram here, the one that's going to be affected is written in italics. So you see my pointer's a little off, so I get off on here. So the C is the one that's going to be changed in there. So we go down, and where it should be AUG, it is. That's my codon. Where it should be CGU, okay, it is here. This is the correct form. And then so on down the line. We're not going to do all of those because those don't get affected. There's the mutation right there. So AUG codes for methionine, we're good. CGU codes for arginine, we're good. And threonine and then leucine on down the line. This is the correct code that it should be. But if this C gets substituted with something like a T, now this is all correct going this way, and so are these guys because they didn't get affected. So all three of these are fine. The one that gets wrong is you bring in you have the wrong messenger RNA right there. So where I should have had CGU, now I have CAU. And CAU codes for different amino acids. So I have histidine instead of arginine. And it seems like it's minor, but again, it can be the difference in a huge shape change in the protein. But you still get a protein. It might not be a good protein. It might not be truly functioning protein, but in sickle cell, you do at least get a blood cell. In something like an insertion or deletion, you may not even get a blood cell. So that would be much more devastating on the developing fetus at that point because they wouldn't be able to make blood, right? So insertion, deletion. What we refer to these is a frame shift mutation. And the reason it's a frame shift is I'm reading in groups of 3 in a certain frame, but as soon as I insert one or I delete one, your mechanism for reading the codons always says reads in group of 3. It doesn't say reads in groups of 3, but if I add one, skip it, or if I delete one, go ahead and just ignore that or substitute it in. It just says read in groups of 3. So it's every 3 it's going to read it. So once again, I'm doing the right thing in the first one. But here we have where I had TAC and then GCA. Now I have insertion. A T was inserted into that one. So where it should have been T, A, and there's the C right there, now I have a T inserted. So it's going to read that group. And then where I should have GCA, that last C got attached, and it can only read in groups of 3. So it shifts everything down the line. So it's not reading it in the correct order. So not only is the first one screwed up, the second one screwed up, and everything down the line is screwed up because it has shifted my reading frame. It has to read in groups of 3. Here's an example of a deletion where they formed, in this example, a sentence for you. The fat cat ate the rat. Again, groups of 3 because codons are in groups of 3, but in this case, we're using letters that make a sentence. So the cat apparently it's a tongue twister too. The fat cat ate the rat. That is a sentence that you can see, you can read, and it makes sense to you. That's how a protein needs to do to your cells. It needs to make sense. But if I delete one letter, I still have to read in groups of three. It's not like I say TE and then I go on and everything else is the same, fat cat. No, I have to read in groups of three. So when I delete that H, the F gets attached to that codon and then down the line. So now I have, this is my sentence. And that is unreadable. That's what we call junk. And that's what you end up making is a junk protein. So either an insertion or a deletion, you end up with a completely junk protein, not a protein that has one error in it or one substitution. It's utter junk. There's nothing redeemable about that protein, and it can't be used for the cell. So those are even more devastating than a substitution would be. And a substitution by itself is devastating. So we want to avoid that at all possible. And like I said, DNA polymerase will self-check during replication to keep that from happening. But then we have these other enzymes that come in. So we have this group of enzymes called repair enzymes, and they go back in and they proofread and they look at DNA if it's been damaged by UV radiation like sunlight in your skin. So they're constantly kind of looking around. And when they see an error, they cut that error out and they replace it with a good one. In this case, we have these, what we call a thymine dimer. And a thymine dimer, you should have T bonded with A, right, across here. So this T with this A, for some reason, with a little bit of UV radiation and a little bit of damage, those T's like to bond together. So they come together and they form a bond, and it makes this bulge. And that's an error. That's wrong. So the repair enzymes can go in, cut that section out, and notice they cut a little bit more out than they need to, but we want to be safe. And then they add in a whole new section so that you're back to good shape again at the end. So we do have several mechanisms to repair these errors that can occur. And you want that to occur because otherwise you would actually accumulate a lot more errors than you do in your lifetime. Having said that, you guys are all little mutants. You have errors. But if it's in a part of the DNA that's not being used, it's not going to affect you in the slightest. Remember, not all your DNA is used in every cell. Your stomach cells are busy building hydrochloric acid. They only need to use the genes for that. So if you get an error in something that says, here's how you build the hair, and it's in your stomach, that gene's shut down. It really doesn't matter. It's never being used anyway, right? If you get an error that is in your hair that should be producing mucus for your nasal cavity or something, whatever it is, and that one's an error, it really doesn't affect it because it's not being used. So you can accumulate some errors without problems. The problem is when we accumulate mutations and we accumulate errors and they lead to something like cancer. So you can have neutral mutations and you can have bad mutations. And we get those over a period of years. So the longer you live, the more likely you're going to have some errors. All right, now here's another problem. Remember that our enzyme pathways, so we had A leads to B, and you had one enzyme that did that. Then B leads to C, and you had a second enzyme that did that. So we have these metabolic pathways, right? And I told you that if one of these enzymes goes wrong, then I build up too much here, and I never get the product over here. That is going to be a problem because I never get my product and I get too much of something else. But if I get an error here, then I get too much of this one and I still don't get the product. So we'll never get the product, but we're getting too much of something else. And it just depends on where the error is along that pathway. That leads to a multiple type of disorders. These three right here, PKU, alkaptonuria, and albinism, all are three genetically linked and genetically inherited disorders of varying degrees of what's going on with the individual. All of them are on the exact same pathway. So if this is my pathway right here, they're all on that pathway. But PKU is the problem with one enzyme, where albinism would be the problem with a different enzyme. So we get a different buildup each time of a certain type of amino acid, and it leads to different outcomes, different problems. And alkaptonuria would be somewhere else on that pathway. When you look at PKU is fairly tragic, but there's an easy fix. PKU stands for fetal ketonuria, and it is the inability to metabolize a amino acid called phenylalanine. And phenylalanine is really important in protein structure. You start getting phenylalanine in your diet immediately from birth because it's in breast milk and it's in the formulas that we feed our children. At first, these children start out normal, but as they are unable to break down excess phenylalanine, it builds up in their systems. Where we get it, we use what we need. People who don't have PKU use what we need, and then we break down any excess. They use what they need, and then they accumulate the excess, and then they accumulate the excess, and they accumulate the excess. And what you end up with is mental disabilities in these individuals. So brain damage that leads to severe mental disabilities in these children. They don't start out that way. They end up that way because of this buildup of phenylalanine. So what we've done as in our healthcare system, every child, when they're born in a hospital or shortly thereafter, if they're born at home, when they go to a hospital for their checkups, they get a heel stick, and we take a little blood, and we can do a genetic test on it. And if they test positive for PKU, all you have to do is say, don't give them any phenylalanine. Sounds easy, but it's not that easy. That means no breast milk, and that means a special formula. And you got to remember, these children still have to grow. They still have to build muscle and bone and all of that. So they need protein. You're going to have to give them protein, but you just have to give them a very specified amount of protein that doesn't have phenylalanine in it. And unfortunately, phenylalanine's everywhere. Like I said, they're exposed from birth, and they're exposed all through their life. But as long as you give them a diet that involves a formula their entire life that supplements all amino acids except phenylalanine, they'll grow normally and have a normal life. But it's a lifelong commitment. If they at 25 decide to go off the diet, say, look, I'm tired, I would like to have a beer and pizza with my friends and a whole bunch of it, and they go off the wagon there, they're going to build up that phenylalanine, and it will demonstrate as brain damage. So it's something that they have to continue their entire life. And it does really become a family commitment too. It's not like you can say, hey, mom and dad and your brother are going to have a pizza. You have to have your special formula and maybe some lettuce or something. The whole family is committed to that. Just to give you an idea, that the diets are very specified. They do have to lay out their diet every day. What am I going to eat for each meal, for each snack and stuff? And it goes down to, if you look it up on the internet, you can say 5 potato chips today. 1 chicken drumstick is more phenylalanine than these kids are allowed in a day, or these adults are allowed in a day. It has more phenylalanine than is good for them. So that's the kind of control they have to have over their diet. But this outcome of the mental disability is entirely avoidable, and they would have a normal development without that phenylalanine. Alkaptonuria is another disorder that's along the same pathway. So it's along this pathway, but it involves a different enzyme. This is urine that you see here. So in A is normal urine. In B is a true indicator that this person has alkaptonuria. They're not metabolizing correctly, and that, again, can lead up to cellular death if it's not taken care of. And then albinism is the inability to produce melanin, and we found out that melanin production pathway is, again, the same pathway that these other disorders, but it's a different enzyme along the way. Porphyria is actually not on this pathway. I included it because it is a genetic disorder that has several outcomes from, and it is on a different metabolic pathway. But porphyria is one where the individuals, if they inherit this gene, and the problems with the proteins is they have very intense light sensitivity. These are burns on his hand from exposure to sunlight, just a little bit of exposure to sunlight, and they get severe burns on their hands. They have reddish types of lips, and gums tend to look red. They tend to have problems with their sanity as they get older. So there's this buildup of different types of chemicals in their neurological system, and so they tend to go crazy. Crazy King George II from England had porphyria, and he showed those symptoms. And the other thing is garlic tends to make it worse. So this misunderstanding about our genetic disorders actually led to a common myth in our society, the vampire myth. It was actually this tragic genetic disorder, or one of the things that led to it. There's a bunch of other things like rabies and stuff that build together with this, but this redness of the mouth, this insanity where this person probably thought they were really bloodthirsty or were going after somebody else, this inability to be in the sunlight, you have to only go out at night. All of that leads into and feeds into this fear that individuals have, and that leads to the vampire myths and stuff. All right, so here is my pathway. Here's the start, and here's the end. So whatever, all the way down here. As we go down, remember each arrow is going to represent an enzyme. So my first one is this protein that converts into phenylalanine. So on my first protein right there. If we have an issue right here, block, boom, phenylalanine cannot be converted to tyrosine. I just blocked this pathway. Not only am I not going to get rid of all of that in carbon dioxide and water, I'm going to build up here, and that's where you get phenylketonuria. If, on the other hand, that one's okay, and we go from here to here, and we get down to tyrosine, and we're trying to get here, I can have a couple blocks along here. I can either block here, and it leads to an inability to make melanin, so that's albinism, or I can have a block that they show right here, which leads to this thing called cretinism, and that is where the brain and the skull don't form to the correct sizes, so that the head is too small and the brain is too small, and that's obviously going to lead to some mental disabilities there, and developmental disabilities, because the brain's controlling all of that. If that's okay, so that part of the pathway is okay, and we get down here and we're going phenylalanine and tyrosine to hydroxyphenylpyrrolic acid, but then that one's blocked, we can get something called tyrosinosis. If that one's okay and we're good, everything's good, we're moving all along, but this one's blocked, then that's alkaptonuria. So you can see that you could have an issue with any enzyme along the pathway. Most of us have all of our enzymes that are being built correctly, and we end up along the right thing. But if you inherit any disorder somewhere along, or a genetic mutation along the way, you can have one of those disorders. You're not going to have all of them. It just depends on where the block is as to what proteins are going to build up in your body and what your outcome is going to be. In case you're curious, we talked about metabolism. So there is online, you can look up the structure of every protein that's out there. We have a human protein project. We have a human metabolome project. So you can look at the metabolome. You can have the human genome project. So you can look up the genome if you're really, really curious and you just want to see a gene and the thousands of ATCG sequences. It's free access, and you can go look it up at any point that you want to. I sometimes have my high school kids go look up a gene. They'll pick out a gene and they compare it from one organism to another organism to see what makes, a rabbit different from a snake or something along those lines. And they can find that same protein that's found in both of them and see where the differences are. How many mutations were there that made them different along the line? And that would be the last of it. So good luck with this unit, and please do a lot of studying for this and practicing.